Experiment 2. Aim: to identify different types of blood cells from a human blood smear when observed under the microscope. Materials required: microscope, permanent slides of a blood smear and chart showing blood cells. Theory: blood is a fluid connective tissue present in the human body. It flows throughout the body in blood vessels. It is an important part of the circulatory system. It consists of the plasma, which is the liquid part of the blood, and the blood corpuscles. The blood corpuscles comprise about 40% to 45% of the blood. Plasma consists of proteins, glucose, fatty acids, hormones, antibodies, and some vitamins. The blood corpuscles or cells are of different kinds and perform different functions. Procedure: the permanent slide is taken and cleaned properly. It is fixed under the low power of the microscope. The slide is then observed under the high power. The cells are identified with the help of the chart. Correct sketches are then drawn with the help of the chart and labeled properly. Observations: the following types of blood cells are seen on observing the blood smear. Erythrocytes or red blood corpuscles, RBCs. These are round and disc-like with a biconcave surface. They do not have a nucleus. They contain hemoglobin, so they appear red. Each RBC has a diameter of about 7.5 mu per meter. Leukocytes or white blood corpuscles, WBCs. These are larger than the red blood cells. They have a nucleus. They do not have hemoglobin, hence they are called white blood cells. White blood cells are of two types. Granulocytes. These cells have granules in their cytoplasm, which can be stained with different types of stains. They are of different types. Eosinophils. They are also called acidophils. They have large granules which are stained with acid dyes and are bilobed nucleus. Basophils. They have few granules and are stained with basic dyes. Nucleus has 2 or more lobes. It is often shaped like an S. Neutrophils. They have a number of granules and they are stained with neutral dyes. The nucleus of these cells has a number of lobes. Agranulocytes. These cells do not have granules in their cytoplasm. They are of two types. Lymphocytes. These are smaller in size as compared to other WBC. They have a large round nucleus and little cytoplasm. Monocytes. These are large-sized cells, about 15 mu per meter in diameter. They have a bean-shaped nucleus. Thrombocytes or blood platelets. These cells are very small. They are almost spherical in shape. They do not have a nucleus. Their size is about 3 mu meter diameter. Number and function of the cells. Red blood cells. RBCs help in transporting oxygen in the body. The lifespan of the RBCs is about 120 days. There are about 5 million to 5.5 million of RBCs per millimeter of blood. Their number is much larger than WBCs. White blood cells. These cells help in fighting disease-causing organisms and constitute the immunity system of the body. They are about 5,000 to 11,000 WBCs per millimeter of blood. Granulocytes, eosinophils. They are 1% to 5% of the total WBC count. They are responsible for antigen-antibody reaction. Basophils. They are 0.5% to 2% of the total WBC count. They are related to allergy condition in the body. Neutrophils. They are present in maximum number, that is, 50 to 80% of the total leukocytes. They kill disease-causing microbe by phagocytosis. Agranulocytes, lymphocytes. They have a diameter of about 15 μ. They are 20% to 40% of the total WBC count. They help in the formation of antibodies. Monocytes. They are 2% to 10% of the total WBC count. They help in destroying pathogens by phagocytosis. The total number of WCs in a given volume of blood is called TLC, total leukocyte count. The number of different WBCs is called DLC, differential leukocyte count. The ratio of the WBCs to the RBCs is 1:600. Thrombocytes. Their number is about 2.5 to 5 lakhs per millimeter raised to the power -3 of blood. Thrombocytes help in the clotting of blood.